Hello again, everyone. My name is Nate. I am the website MCAT. Today's video is going to be everything that you need to know for the MCAT about glycogenesis. As per usual, I've made notes that accompany all of the information on these slides, so I highly recommend checking out the link in the description so that you can go download those notes. You can make a copy of them and add to them or whatever you want, so that all you have to do in this video is just sit back and listen, make sure everything I'm saying is making sense. It's also where you can find my other resources. If you like the way that I teach, you can also suggest ideas for other videos, other cheat sheets, anything like that. You can find all of that on my website. If you are interested in tutoring, especially for things like cars as well, I offer that through my website as well. So just as a reminder of where we're picking up with glycogenesis, if you're not watching these videos in a linear order or anything like that, we're talking about the postprandial state right now, where you've just eaten a meal, you've got a ton of glucose, our body's trying to use that glucose for whatever we can. So glycogenesis is actually going to be picking up at this portion of the postprandial state where we've been utilizing all of this glucose and we are now at a point where we have enough ATP. We have enough ATP and there's still extra glucose. Glycogenesis, or the production of glycogen, is going to be storing this glucose as a molecule called glycogen. So if our cells have a high amount of ATP, we don't have any reason to keep running aerobic respiration, to keep utilizing that glucose through aerobic respiration. So then we're going to divert all of that glucose and store it as glycogen in that process called glycogenesis. We're going to talk about this in a separate video, but the liver and fat cells might actually have some extra acetyl-CoA laying around, and acetyl-CoA is going to be stored as fatty acids. So glucose is stored as glycogen, acetyl-CoA is stored as fatty acids. That's going to be the easier way to think about it on the MCAT. That's a process called fatty acid synthesis. We're going to talk about that in our next video. So I want to bring up that glucose transporter GLUT2 again. GLUT2 is on the liver that is activated when there's a ton of glucose in the blood. Basically, this is just a sign that we have excess glucose and that we can start storing some of it. Whenever GLUT2 is activated, the liver can take some of that glucose that it's pulling in and actually start to store it as glycogen. So GLUT2 being activated is one of those signals that we can start saving glucose as glycogen. It's also going to signal the further production of insulin from the pancreas to keep removing glucose from the blood. Going to get our cells to keep utilizing that glucose until glucose levels start to return closer to normal levels. If you eat something with a whole bunch of carbs in it, basically just whenever you eat a meal, especially something with a lot of carbs in it, I'm thinking about the rolls at Texas Roadhouse right now that come with cinnamon butter. Probably going to order those right now that I've mentioned that. But after you've eaten that, you're going to have a whole bunch of glucose floating around your blood. Your pancreas is going to swarm the blood with insulin, and that insulin is going to start doing two different things. First, of course, it's going to cause that glucose to get sent into the cells all over the body so that they can start making ATP with it. But additionally, insulin is also going to help us start glycogenesis, especially in the liver. We're going to use glucose one way or another. We're either going to produce ATP with it or we're going to store it if it's in excess. So how do we store glucose, right? Of course, as I've been mentioning, it's as a polymer, specifically a polysaccharide called glycogen, within mammals at least. We're talking about mammalian metabolism. We're a little bit selfish on the MCAT in that regard. So glycogen is the molecule that we're going to be talking about. So glycogen is a polysaccharide that essentially is just a whole bunch of glucose monomers that are strung together. It's specifically within mammals, and it's going to be used as what we call short-term storage. Basically, it's the short-term storage of glucose to be utilized within 24 hours. Our glycogen stores are constantly going to vary. We're constantly going to be building it whenever we've eaten a meal, and we're constantly going to be using it whenever we are starting that short-term storage, like whenever you go to bed. So glycogen is going to be a really quick way for us to store glucose for short-term storage. It's going to utilize two types of bonds that we're going to talk about that are important for the MCAT. It's going to utilize an alpha-1,4 glycosidic linkage. This is going to build the linear portion of glycogen, as well as an alpha-1,6 glycosidic linkage. This is going to form the branch points that kind of stick off the main glycogen chain. So glycogenesis does require the input of some ATP, but that's perfectly fine. We are at a point, like we've been talking about in the postprandial state, that we've been running aerobic respiration a whole bunch. We've produced a whole bunch of ATP that we don't really need any more of it. So we're going to take this glucose, we're going to use a little bit of that ATP to start storing that glucose as glycogen in this high ATP and high glucose condition. So aerobic respiration, which is going to be a catabolic process, is going to produce energy that we're going to store in ATP. Glycogenesis, however, is an anabolic process where we're building a molecule. This is going to use energy. In general, breaking down molecules such as aerobic respiration will produce energy. Building molecules such as glycogenesis and fatty acid synthesis, that's going to require energy. So glycogenesis requires ATP, but that's okay. So to give our brief overview of glycogenesis, it's going to be occurring in the cytosol, meaning it's a relatively oxygen-free reaction, mainly within the liver cells, because the liver can pretty much do anything that it wants, as well as the myocytes, basically muscle cells. Muscle cells are going to be capable of doing glycogenesis as well. For the MCAT, those are pretty much the only two types of tissues within the body that are going to be doing it. And the goal of glycogenesis, of course, is to store glucose as glycogen, the short-term storage molecule for glucose. Just to help out with the naming, glyco, you can think of that as meaning glycogen, and genesis means the production of. The genesis of glycogen, the production of glycogen. Essentially, within this reaction, we are taking G6P, which of course is the second molecule in glycolysis, comes right after glucose. We're going to be taking that G6P, we're going to be using some ATP to turn it into glycogen. So of course, we're going to be using some ATP to turn glucose into G6P via the enzyme gluco and hexokinase. We're going to talk about all of these steps on the next slide. We're going to take this G6P and then turn it into G1P. And once we form this G1P, we're going to be then linking a whole bunch of G1Ps together to form this linear chain of glycogen. Again, that's just a brief overview. It may not fully make sense right now. We're going to talk about the steps in more depth on the next slide. So of course, when are we going to be doing this? When we have a whole bunch of glucose left over and we already have high ATP levels. Of course, this is going to be occurring during that postprandial state. You've just eaten a meal where we're going to be using that meal to produce ATP. And once we've done so, we're going to store the rest of it. So of course, because of that, the hormone that's going to be promoting it, just like the rest of the postprandial state, is going to be insulin. It's also going to be inhibited by glycogen. There's 4 enzymes that you need to know: gluco and hexokinase, phosphoglucomutase, glycogen synthase, and branching enzyme. We're going to talk about those on the next slide. So just to explain how glycogenesis works, of course, the liver and muscle cells, because GLUT2 has been activated in this high glucose condition, is going to move that glucose into the cytosol of those cells. So we have our glucose molecule up there in the top left corner. I've labeled the carbon numbers. We have carbon 1 on the right, carbon 4 on the left, carbon 6 at the top. That's going to help with understanding the naming of the bonds that we're going to be forming here. So as per usual, gluco or hexokinase is still going to convert that glucose into G6P. This is technically the first step of glycolysis, but we're still going to be utilizing this in glycogenesis. We need this G6P molecule in order to actually start making glycogen. So remember when we talked about glycolysis, that the actual committed or rate limiting step of glycolysis is not until we get to PFK, which is a little bit later. As you can see here, we can send G6P through the rest of glycolysis if we wanted to, but we can also divert it and send it through glycogenesis. That's why this enzymatic step is not considered a committed step. It's not a guarantee that we're going to be producing glycogen in glycogenesis or pyruvate in glycolysis. Of course, this step is going to utilize ATP. Like I've mentioned earlier, glycogenesis is going to use ATP, but that's okay because we have plenty of it, where we're basically just sticking a phosphate onto that 6th carbon. Like I've mentioned earlier, we could potentially just send this G6P through the rest of glycolysis. We're specifically going to be doing this in low ATP conditions, but in high ATP conditions, this is when the liver and muscle cells are going to start using the enzyme phosphoglucomutase. This is an enzyme that's reversible. Notice that the arrows go both direction here. That it's going to be diverting G6P from glycolysis, and it's basically going to flip that phosphate from the number 6 carbon to the number 1 carbon. I've tried to highlight in yellow, or with a yellow circle, I guess, the thing that changed or the bond that formed. That's the thing that you can be looking out for on this slide as we keep going through all of these enzymatic steps. So now that the phosphate is in this 1st carbon position, that's actually going to make our polymerization steps a lot easier, because we're going to be extending this glucose chain off of this number 1 carbon. The next enzyme is going to take this G1P and it is going to cleave off this phosphate. This is done by the enzyme glycogen synthase, which by cleaving off this phosphate, that is going to provide energy to form the alpha 1,4 glycosidic linkage. We've linked carbon 1 of the first glucose monomer to carbon 4 of the next one. Glycogen synthase is going to be forming these alpha 1,4 bonds. For the purposes of the MCAT, the alpha 1,4 linkage, that is the linkage that glycogen synthase is making. So glycogen synthase can just keep repeating itself over and over again to get this really long linear strand of glycogen. But we can also branch off of this growing linear strand. This is done by an enzyme called branching enzyme. It's an enzyme that's going to create what's called an alpha 1,6 bond. Notice in this new yellow circle down at the bottom of the page that we have linked carbon 6 of a glucose in the linear chain to carbon 1 of a new glucose that's kind of branching off of that main glycogen chain. So branching enzyme is the only enzyme on the MCAT that is going to be capable of forming these alpha 1,6 bonds. Glycogen synthase is going to be forming all of our alpha 1,4 bonds. Branching enzyme is going to be forming all of our alpha 1,6 bonds. So branching enzyme is going to keep forming a whole bunch of these different branch points. And once it's formed that alpha 1,6 bond, it's stuck that one extra glucose on as a branching point, it's done. But now that we've formed this alpha 1,6 bond, this branching point, we can actually have glycogen synthase come in and add another glucose onto this branched glucose. Looks something like this. So we can keep forming kind of like a tree structure where glycogen is going to keep growing down the parent chain. That is done by glycogen synthase. Branching enzyme is going to form these little branching points, these little forks in the road. And once we formed that fork in the road, we can actually send glycogen synthase back in to keep extending off of those new branch points. So really important to understand, branching enzyme is only making that first alpha 1,6 bond. Any other alpha 1,4 bonds after that are going to be done by glycogen synthase. There are going to be other steps in glycogenesis, some of which also require ATP. You just don't need to know them for the MCAT. Luckily, if you understand every step we've talked about so far, you understand glycogenesis. Glycogenesis doesn't necessarily have a stopping point. There isn't one structure of glycogen. It's basically just a really big polysaccharide of glucose that we're just going to keep adding on to as needed. So from here, glycogen is then going to be stored as a short-term reserve as glucose. It's really easy for us to basically just start chopping off each one of these glucose monomers in a process called glycogenolysis. We're going to talk about that in a different series of videos whenever we start talking about the short-term fasting or post-absorptive state, that we can basically just store this glycogen for those later times when glucose levels start to drop. It's been a while since you've eaten a meal. Your glucose levels are going to start to go away. We need glucose, and glycogen serves as that reserve for that glucose. So in this postprandial state where we've eaten a meal and we've got a ton of this glucose floating around, we're building this glycogen so that it can be utilized later on. So that was glycogenesis for the MCAT. Nothing more, nothing less, only the specific things that you need to know. As always, you can click the link in the description for my other free resources, suggest video ideas, find my tutoring, yada, 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 so on and so forth. You can also find the notes that supported these slides in the description in case you already missed that. All of that can be found at the links in the description.